My name is Associate Professor Mirana Ramialison, and I'm the head of the Developmental Systems Biology Laboratory at the Australian Regenerative Medicine Institute. Today I'll be speaking to you about my passion, which is DNA, and I would like to talk to you about the secret life of, more specifically, junk DNA, and we'll come to that. So DNA is my passion, and we owe this field thanks to the discovery of Watson and Crick in the 1960s, but mostly as well to a pioneering woman, which is Rosalind Franklin, who actually contributed to that discovery, but unfortunately was not awarded a Nobel Prize because of her early death. But without these three people, we wouldn't know much about DNA today. And as we all agree, DNA is the basis of life. So the DNA, in a sense, what is it? It's just a succession of letters that actually compose the message and the information that will give rise to our cells, and also compose the information that will give rise to our individuality. But it's still fascinating to me that at the end of the day, it's just a succession of letters, A, C, G, and T. So we've learned a lot about the DNA, and we know now that the DNA contains genes. And so we agree, genes are the essential component of the genetic message. Genes are easily identifiable in the DNA because there is a known genetic code that has been unraveled. And this genetic code basically translates the information from the DNA into genes, which then encode proteins will make up our cells and our bodies. Genes are a very important component of our DNA. And we know genes are essential for everything. And I also have another passion, which is organ formation and how, our bodies are formed the way they are. So genes are these essential components that are crucial for the formation of organs. What I'm showing you here is a fruit fly. And what I would like you to pay attention to is the eye of the fruit fly. And I'm going to show you known examples in literature that show the importance of genes. So this is a fly where you can see the eye here, and we have a gene in the flies which is called eyeless. And if this gene is removed, or if the flies lose it, you can see here that the fly doesn't have eyes anymore. On the contrary, if this gene is introduced and this, as we call it, the gene is gained, then the fly can have eyes in places where it shouldn't have. For example, you can see here that the fly has eyes on the legs as well. I think this example really shows how genes are important. The genes are the molecular information that dictate the formation of organs, and having a gene or losing it is actually very important for our bodies. And a lot of genetic diseases occur because we have mutation in our genes, and the whole field of research around molecular biology and genetic information is to understand what are the genes and what do they do, so we can understand the genetic components that are essential for formation of the body. So let's go back. So genes are a succession of letters, A, C, G, T, and then we've been working on the premise that if we find all of the genes that compose our genomes, would we be able to then understand the secret of life? Because if we know what are all the genes that compose our bodies, we can understand how the body forms, how the cell function, how disease occurs, because then if we find all the genes and find what they do, we can understand what happens when something goes wrong with these genes. So therefore, it means we can understand what happens in disease. So this is a very important concept where finding all genes has been really the holy grail into understanding life. And finding all genes means we need to know exactly what's in our DNA. What are the letters that compose our DNA? Finding those sequences and understanding them is really fundamental to our understanding of the genetic information and ultimately understanding our bodies. So the sequencing of the DNA has been achieved in 2001. The first draft of the human genome has been published already back, 20 years ago. And what have we learned since then? Well, we've learned that we have 25,000 odd genes in the genome, give or take. So we know exactly now in the human genome what are the genes. We can find them in the genome and we know exactly where they are. But what was really interesting is that the genes, they actually only compose 2% of our DNA. So we're working on the premise that genes are the secret of life. We identified all the genes in our genome and realized when we sequenced the genomes that actually only 2% of our DNA is composed of genes. This has been the question that fascinates me since the beginning of my career. What is the rest of the DNA doing? And this is a question that's still largely unresolved nowadays. So the main question is 98% of DNA junk? So the first sequencing of the human genome happened in 2001, but as I mentioned, until a few years ago, it's been widely reported that maybe the rest of our DNA, which is 98% of it, doesn't encode the gene, which means that this DNA, the rest of the DNA, is garbage. Aren't genes everything? So what is this 98% of DNA is non-coding, is doing? We call it non-coding DNA because it doesn't encode for genes, it doesn't encode for protein. So this is my field of research I'm really passionate about. I don't believe that 98% of our DNA is junk. So is there any function of the junk DNA that we haven't uncovered yet? And so there's a large body of research now trying to understand the function of junk DNA. And when you think of this example, it's really, I guess, telling us what is going on behind the genes that we still have to uncover. So I showed you this example to show that when the gene is absent, we don't have eyes. When the gene is present, we have more eyes than we need. But ultimately, in a normal body, what controls the gene to it be expressed where the gene should be expressed? In other words, when the gene is present, it does its job. But what tells the gene to be present where it should be present? So at the end of the day, we still have not resolved what controls the presence or the absence of the gene. We know what the gene does when it's present and when it's absent, but we still don't know what controls the expression of the gene. In other words, what turns the gene on in some places and off in other places. So this is the new field that we're working on. So this representation is what we thought about the genome before at the onset of its sequencing and before we understand more about the junk DNA. So in pink here, you have a representation of a gene, which in the whole picture represent only 2% of our DNA. And the rest of it is supposed to be junk or, with no function. But in fact, the realistic picture now, after years of work, is that we and others have uncovered that the rest of the genome is actually full of information. And all this information is aimed at understanding and controlling the expression of the genes. So this new information that is encoded in the part of the DNA that doesn't encode for genes is called regulatory DNA, which is in essence all the information needed to switch genes on or off. So it's been hard to uncover this information because unlike genes, they don't have a specific genetic code. This regulatory DNA doesn't have a specific information that can be encoded and then translated easily. So that's why it's been very difficult to understand the function of this, what we called before junk DNA. So my field of research is regulatory DNA. We also call these cis-regulatory sequences. We also call them enhancers because they enhance the expression of the genes, promoters because they are needed to promote the expression of the genes. They're also called silencers because sometimes as well they shut down the expression of the gene. So my job is basically to find switches that for genes that will turn the genes on or off. So how do we do that? As mentioned before, we have basically 98% of our DNA, which was considered to be junk. And we know now that this 98% contains switches, information that are really important for genes. But the DNA is composed of 3 billion letters. So how do we find this critical information in absence of a genetic code? So my laboratory is dedicated to design new tools to find these needles in the haystack. What we're trying to find is these specific sequences of letters that we call switches that will be able to turn genes on or off. And the field we were working on is called bioinformatics, because we are designing novel program and novel software to try to decode the DNA in order to predict where these switches are and understand then how genes are regulated. Here's an example of one of the tools that we've generated in my laboratory. It is called Trawler, as in it trawls through the genome to try to find and identify these specific switches that will be able to control the expression of genes. The software is designed for many different species, not just human, so we can run our search and our prediction on a large range of organisms. This is, for example, the output of our software Trawler. So if you insert, if you're interested into a genome of interest and try to identify whether there are switches or little words in our genome that are most important, that occur often, that could encode for an information to regulate the expression of the genes, Trawler will predict and tell you, well, in your data set, this is the little switch that I found. This is a little bit of information that I found that will help you to understand that this sequence is important for regulating genes or not. So in my laboratory, we also validate this prediction, and this is an example of one of the prediction that we generated before. So each of these color dots basically represent switches that our software has predicted. So these switches, again, are little bits and pieces of letters that are present in the whole genome. And as you can see, they're only about 6 letters long, and the genome is composed of 3 billion of these letters. So it's literally looking for needles in a haystack. And one of these color dots represents one of these sequences. And what we've done in the lab to really show that these are actually switches, what we do is that we put all of these switches in the animal, and you can see here in green, for example, that the gene is turned on because it's turned green. And when you remove those switches, the gene is absent. So we don't see the green anymore, which really tells you that these switches are able to turn on or off the expression of the genes. So these switches are found in our genome, and they are actually really the one that controlling the expression of our genes. So where we want to go in the future, we still need to find a lot about these switches. And one very important and emergent aspect is that we want to look at this information in 3 dimensions. Why is that? Our body are not flat. We are working with 3-dimensional organs. So all of the research so far was performed only on information obtained in 2 dimensions or obtained from cells that have been put together. And we often lose the spatial information from where do the genes come from and where do we actually interrogate the cells to find that genetic information. So we think that in the future, all of the analysis that have been done before will be projected into 3 dimensions. And this is where my laboratory is focusing on now, generating new tools so we can understand our genes and our genomes in 3 dimensions. So the last tool I would like to introduce to you is called 3D CardioMix. So this tool is basically a software where we have looked at the heart and looked at all of the 25,000 genes that are present in the heart. And not only we'll look at it, just having a list of numbers and genes, we actually look in a 3-dimensional space, where are these genes expressed in the heart? So now we are able to look at this information in 3 dimensions and not anymore in 2 dimensions has been done before. So we think this is going to be the next challenge and the next exciting opportunity in the coming years, that we'll be able to look at genomic information and genetic information in 3D dimensions. So with that, I'd like to finish then to summarize. Is most of our DNA garbage? Certainly not. We know that 98% of the DNA is not encoding for genes, but definitely within that DNA, there's still a lot of information, and more specifically regulatory DNA or switches that are really important for the regulation of our genes. So this quote is the summary of my talk. Much of what has been called junk DNA in the human genome is actually a massive control panel with millions of switches regulating the activity of our genes. And my whole job is to find more and more of these switches so we can complete the panel, so we can understand how our body are formed, and also we can understand when we have a disease, which switches actually malfunction and didn't work very well. With that, thank you very much. And I really would like to acknowledge my team, past and present members. They're the one behind all of this work, and also my institute, Monash University, and my national and international past and present collaborators, and the funding without which we cannot do this research. Thank you.